DNA replication is the process which new strand of DNA is produced from the parent DNA. When two strands are separated, each strand acts as the template for the formation of the new strand. There are three steps of the DNA replication, which are initiation, elongation, and termination.
There are three models of DNA replication. In the semi-conservative model, the two parental strands separate and each makes a copy of itself. After one round of replication, the two daughter molecules each comprises one old and one new strand. In the conservative model, the parental molecule direct synthesis of an entirely new double-stranded molecule such that after one round of replication, one molecule is conserved as two old strands. This is repeated in the second round. In the dispersive models, material in the two parental strands is distributed more or less randomly between two daughter molecules. In the model shown here, all material is distributed symmetrically between the two daughter molecules. Other distributions are possible.
DNA replication is initiated by the helices. Helices unwind the parental double helix and forms the replication forks. Then, the single-strand binding proteins stabilize the unbound parental DNA. The leading strand is synthesized continuously in the 5' to 3' direction by the DNA polymerase. The lagging strand is synthesized discontinuously. Primase synthesize a short RNA primer, which is extended by DNA polymerase to form an Okazaki fragment. After the RNA primer is replaced by DNA ligase joins the Okazaki fragment to the growing of the strand. Replication must be terminated to produce two daughter DNA molecules and to regulate and coordinate replication with cell division.
So I will explain about transcription. So what is transcription? Transcription is a synthesis of RNA molecule using DNA molecule. Only one of DNA strand is transcribed. The enzyme used is RNA polymerase. There are four major types of RNA molecule, which are mRNA, rRNA, tRNA, and snRNA. Transcription divided into three processes, which is initiation, elongation, and termination.
Have you ever had to transcribe something? Maybe someone left a message on your voicemail and you had to write it down on paper? Or maybe you took notes in class then rewrote them neatly to help you review? As these examples show, transcription as a process in which information is rewritten. Transcription is something we do in our everyday lives, and it's also something our cells must do in a more specialized and narrowly defined way in biology. Transcription is the process of copying out the DNA sequence of a gene in the similar alphabet of RNA. Transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as protein. The goal of transcription is to make a RNA copy of a gene's DNA sequence. For a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide protein or protein subunit. Eukaryotic transcription need to go through some processing steps before translation into proteins.
In prokaryotes, the gene involved in transcription basically has three regions. First is a promoter sequence, which attracts RNA polymerase to begin transcription at a site specified by the promoter. Second is the transcribed sequence, called the RNA coding sequence, corresponds with the RNA sequence of the transcript. Last is a terminator region, which is downstream of the RNA coding sequence that specifies where transcription will stop. Transcription process. RNA polymerase binds promoter in two steps. First, it binds to the 3,5 sequence. Second, it binds tightly to the 10 sequence and positions to begin transcription. Once initiation is completed, RNA synthesis begins. After 8-9 ATPs have been joined in the growing RNA chain, sigma factor is released and reused for other initiations. Core enzyme completes the transcription process. Core enzyme untwists DNA helix locally, allowing a small region to denature.
Transcription. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription is performed by enzymes called RNA polymerases, which link nucleotides to form an RNA strand using a DNA strand as a template. Transcription has three stages, which are namely initiation, elongation, and termination. In eukaryotes, RNA molecules must be processed after transcription. They are spliced and have a 5-inch cap and a poly A tail to put on their ends. Transcription is controlled separately for each gene in your genome.
I'm going to explain about translation. In this stage, the mRNA is decoded to build a protein that contains a specific series of amino acids. In translation, there are three steps, which is initiation, elongation, and termination.
At the fifth cap of mRNA, the small 40 subunit of the ribosome binds. For translation, to start the start codon 5' AUG must be recognized. This is a codon specific to the amino acid methionine, the large 60 subunit of the ribosome that binds for elongation to occur. While medtRNA occupies the P site, another amino acyl tRNA with an anticodon complementary to the next codon comes to occupy the A site. This process requires GTP. The enzyme peptidyl transferase forms a peptide bond between methionine and the next amino acyl tRNA. The tRNA molecule in the P site becomes uncharged and leaves the ribosome. The ribosome then translocates along the mRNA molecule to the next codon. This opens up this A site for the next amino acyl tRNA. The polypeptide chain is built up in the direction from the N terminal to the C terminal. The 3 stops codon enters the A site. No tRNA molecules bind to this codon, so the peptide and tRNA in the P site become hydrolyzed, releasing the polypeptide into the cytoplasm. The small and large subunits of the ribosome dissociate, ready for the next round of translation. The polypeptide produced undergoes post-translational modification before becoming the fully active protein.